here at Oracle, we have live classes to elevate your learning experience. Then you also have the availability of recorded lectures. Then apart from that, we also provide you with notes and MCQs based on the latest curriculum. Then a very special feature of Oracle is that here at Oracle, we also provide personalized counseling sessions. So apart from the academic support, if you need any non-academic support, then you can feel free to book a slot with us anytime. We have 3 different subscription plans at Oracle. That is starting from the basic, which is for 3 months and comes at 15,000. The standard plan, which is for 6 months and it costs around 30,000, and the premium plan, which is for 12 months, it is available at 50,000. Then apart from that, you can get a 20% discount by using my promo code as displayed on the screen below. Here. Then briefly introducing you to the internal exam marathon. So this series has been specifically curated to target your university exams, which are approaching recently. So this is based on rapid revision of all important questions and whatever is frequently asked is in the exams. Volatile facts will be dealt, multiple choice questions will also be taken up to assure top-notch grades. Okay, so let's begin with sulfur-containing amino acids. So in the introductory class, as I had already told you, the sulfur-containing amino acids, they include your cysteine and your methionine. So beginning with cysteine. I think there is some audio-video lag. Okay. So your cysteine, it is a non-essential, purely glucogenic polar amino acid, and it answers sulfur tests. So if you remember, while talking about amino acids in the introductory class, I told you that your cysteine, it answers the sulfur test, that is the lead sulfide test, whereas your methionine doesn't. So why so? Because let's have a look at the structure of cysteine here. Because your cysteine, it has a free terminal sulfhydryl group. Okay, so this sulfhydryl group, it can be liberated. This sulfhydryl group, it can be liberated to form lead sulfide. Okay, so therefore your cysteine, it answers your sulfur test because it has a free terminal sulfhydryl group. Okay, and it is purely glucogenic. And polar amino acid, why? Because of this free terminal sulfhydryl group. So this is going to provide some polarity as well, because it is free and terminal. It is an ionizable group. Okay, so that was briefly about the structure of cysteine. But your methionine, on the other hand, it does not answer sulfur test, because if you see here in the structure of methionine, your sulfur here, it is trapped between these two carbons. Your sulfur is not free and it is not present at the terminal end, rather it is engaged in a thioether linkage. Okay, so the sulfur here in methionine, it cannot be liberated easily, and therefore it cannot combine with lead to form lead sulfide, and therefore it does not answer your sulfur test. And it is non-polar for the same reason, because your sulfur is not easily liberated. It is purely glucogenic. Okay, now let's begin with the methionine metabolism. So if you talk about methionine, okay, so beginning with methionine, here the structures are not required. The only knowing the names will solve the purpose, but for the sake of understanding, I've also shown these structures. So see, if you consider the structure of methionine here, as I've already told you, your sulfur, it is in a thioether linkage. It is trapped between two carbons, as you can see. Okay, now what is happening? So your methionine, therefore, since the sulfur is trapped between the two carbons, it is not a methyl donor, okay? Because you see, carbon, sulfur, carbon, okay, and this terminal methyl here. This is the terminal methyl, okay? So what is happening? This bond between the sulfur and the terminal methyl, this is a covalent bond. And covalent bond, we all know that they are very strong bonds. Okay, therefore your methionine cannot be a methyl donor. So the terminal methyl is attached to the sulfur via a covalent bond which is very strong. Therefore, this terminal methyl, it cannot be given to any acceptor. And therefore, methionine is not a methyl donor. So what is happening? This terminal methyl is attached to sulfur via the strong covalent bond, and therefore it cannot be easily liberated, and therefore we say that methionine is not a methyl donor. Okay, now let's see what is happening. So this methionine, it is acted upon by an enzyme which is known as methionine adenosyl transferase. In short, we call it MAT. Okay, here, this enzyme one here. And what is happening? It is an adenosyl transferase. So from this ATP, as you can see here, it is transferring an adenosyl to methionine. So now if you see what is formed, SAM is formed. What is SAM? It is S-adenosyl methionine. So from this ATP, the adenosyl has been transferred to sulfur, you can see, via this bond. Okay, now what happens because of this? Some bond energy is shifted from the terminal methyl towards this. Okay, so now this covalent bond, which was there, now it becomes slightly weakened. Okay, now it has become weakened. Now this methyl can be easily liberated. Okay, so what happens now? Now this SAM is acted upon by another enzyme which is known as methyl transferase. Okay, here this enzyme is methyl transferase, and what happens? This methyl transferase, it will transfer this methyl to an acceptor. Okay, so the methyl will be accepted by an acceptor, and in turn, what is formed, as you can see here, S-adenosyl homocysteine, SAH, that is formed. So you can see this methyl has been here, from here has been transferred to an acceptor, and you left with S-adenosyl homocysteine. Now this S-adenosyl homocysteine, this adenosyl will be removed from it, and you end up with homocysteine. Okay, now this homocysteine, it can have two fates. Either it can go for cysteine synthesis, or it can regenerate your methionine. And for the regeneration, if you consider the structures of homocysteine and methionine, the only difference is in the presence of a methyl group. So if you add a methyl back to homocysteine, it will again become methionine. Okay, so that was about the metabolism of methionine. And also from homocysteine, when you're forming methionine, we'll also talk about this in detail later in the lecture. But for now, just know that you need vitamin B12 for this reaction. Okay. So this is the same thing that I've explained to you. It says that SAM, that is to the sulfur and adenosyl residue, is attached. Okay. So what is S-adenosyl methionine? That is to methionine, to the sulfur of methionine, you attach an adenosyl residue. So you end up with SAM, and the enzyme is MAT. Now what happens? This terminal methyl is liberated and given to an acceptor, and what is formed? SAM is converted to S-adenosyl homocysteine. Now by the removal of adenosyl, S-adenosyl homocysteine converts to homocysteine, and S-adenosyl methionine, that is SAM, it is the principal methyl donor for almost all transmethylation reactions. Therefore, we've already studied previously while studying so many amino acids that we had studied that SAM is the methyl donor, SAM is the methyl donor. Now we know why. Because the methyl, the terminal methyl in methionine, it is bounded covalently with the sulfur and cannot be easily liberated. Okay, so you have to form an intermediate which is known as SAM, and then the bond energy shifts slightly from that covalent bond to the sulfur and adenosine bond, and the covalent bond between the sulfur and the methyl is weakened, and then finally your methyl can be liberated and taken up by an acceptor. So that was about the metabolism of methionine. Now let's have a look at the significance of these reactions. Okay, the significance of SAM, S-adenosine methionine, that is formed. It is involved in all transmethylation reaction. Okay, it is also involved in polyamine synthesis, and it is also involved in DNA methylation. Okay, so it is very important, your transmethylation reactions. If you remember when we were talking about keratinine, remember then also SAM was needed. Then if you remember in epinephrine, norepinephrine synthesis, epinephrine, norepinephrine synthesis, there also you needed SAM for methyl transfer. Then polyamine synthesis. In polyamine synthesis, you also need SAM. What are polyamines? So polyamines, they are organic compounds with more than one amino group. They are organic compounds with more than one amino group. Okay, and so they have more than one amino group, so which makes them positively charged. Okay, so they interact with the negatively charged DNA. So they are positively charged, and we know that DNA is negatively charged, so they interact with DNA. With DNA, and therefore they are playing important role in regulation of gene expression. They have a role in regulation of gene expression. Okay, so and also your polyamines, they are derived from ornithine and methionine. An example is spermidine and spermine. And DNA methylation takes place in order to protect the proteins from degradation. Okay, now talking about the fates of homocysteine. So as I said, the homocysteine that is formed, it can either go for regeneration of methionine, or it can have a glucogenic fate, or it can synthesize cysteine from it. So let's take each of the fates of homocysteine one by one. So first talking about the regeneration of methionine. Okay. So what is happening? You have homocysteine. So this homocysteine, if we compare the structures, okay, I'll show you. If you talk about the structures, this is your carbon. H2. So this is the constant part of any amino acid, and then you have CH2, CH2, sulfur, CH3. So you have this extra terminal methyl. This is the structure of what? This is the structure of methionine. And in homocysteine, this terminal methyl is missing. Okay, so you see, this is the difference. So what you have to do, if you want to convert this homocysteine into methionine, you only have to add this terminal methyl. Okay, so the homocysteine, it has one methyl group less than the methionine. So you only need to add this extra methyl. Now, for this, what is happening? Your methyl donor is methyl tetrahydrofolic acid. Here you can see methyl tetrahydrofolic acid. It But this methyl tetrahydrofolic acid, it cannot give its methyl directly to homocysteine. Okay, so what happens? It is going to give this methyl to cobalamin first and form methyl cobalamin. This here. Okay, methyl cobalamin will be formed, and then this methyl cobalamin will give its methyl to homocysteine, and you end up with methionine. Okay, so what I'm saying is that methyl donor here in this case, in order to convert to homocysteine, this is homocysteine. So homocysteine has one methyl less. So in order to convert homocysteine to methionine, we need to add a methyl, and for this, the methyl donor is methyl tetrahydrofolic acid. But this methyl tetrahydrofolic acid, it cannot directly donate methyl group to homocysteine. So first it is going to give it to cobalamin to form methyl cobalamin. Okay, now this methyl cobalamin, it is going to donate the methyl to homocysteine to form methionine. Okay, so now let's talk about in case of deficiency of B12, that is your cobalamin, what happens? So methyl tetrahydrofolic acid cannot be converted to tetrahydrofolic acid. So obviously your methyl tetrahydrofolic acid, it was donating its methyl to cobalamin to form methyl cobalamin. But what is happening? There is deficiency of cobalamin. So now methyl tetrahydrofolic acid, it cannot give its methyl to cobalamin, and therefore it cannot be converted to tetrahydrofolic acid. Free tetrahydrofolic acid will be decreased. Now this free tetrahydrofolic acid is very important because it plays important role in one carbon metabolism. For example, in thymidate synthase, where thymine is synthesized. Okay, now what is happening? So there is not an actual deficiency of tetrahydrofolic acid, but there is a functional deficiency because of free tetrahydrofolic acid. Okay, you do have tetrahydrofolic acid in the body, but not as free tetrahydrofolic acid. It is present as methyl tetrahydrofolic acid. Now this is known as folate trap or tetrahydrofolic acid starvation. A folate trap affects your DNA synthesis. Why? Because your thymine synthesis is being affected. So this folate trap ends up affecting DNA synthesis, and when DNA synthesis is affected, what do you have? You get megaloblastic anemia. Blastic anemia is seen. Okay, you have large immature abnormal RBCs. We're talking about deficiency of cobalamin acid. Now, homocysteine, it cannot be converted to methionine. Thus, homocysteine accumulates. It is very obviously your homocysteine, it was being converted to methionine. And for this, you needed what? You needed both cobalamin and folic acid. But what is happening now? That there is a block in this conversion. Okay, so what will happen? Your homocysteine will increase because of deficiency of either cobalamin or folic acid. Okay, so homocysteine is not converting into methionine, leading to accumulation of homocysteine. Now increased homocysteine is a risk factor for thrombosis, leading to coronary artery diseases and cerebrovascular accidents. When there is increased homocysteine in the blood, it also leads to excretion of homocysteine in urine. What is homocysteine? It is two homocysteine. They join together via disulfide bond. That is your homocysteine. So two homocysteine joined by disulfide bond. So there will be excretion of homocysteine in the urine. So that was about conversion of homocysteine to methionine. Now having a look at the glucogenic fate of homocysteine. Now this homocysteine here, it combines with serine. This is your serine, as you can see. Okay, so it combines with serine, and the enzyme is cystathionine beta synthase. Here, this enzyme number donated by 5. So this is cystathionine beta synthase. And what is formed? Cystathionine is formed, and for this reaction, the coenzyme is PLP. So what is happening? Your homocysteine, it combines with serine to form cystathionine. This is then acted upon by another enzyme which is known as cystathionase. Here. Now this cystathionase, it will split this cystathionine into homoserine and cysteine. So what is this? This is an example of transsulfuration reaction. Transsulfuration reaction. So what is happening? The sulfhydryl from methionine. This sulfhydryl group here. So this is in present in homocysteine, and this homocysteine has been formed from methionine. So what do we say? That the sulfhydryl from methionine, it is transferred to serine. You can see here in cystathionine to the serine, this sulfhydryl group has been transferred. So we say this is an example of transsulfuration reaction where what is happening? The sulfhydryl group from methionine is transferred to serine to form what? To form your cysteine. Okay. So the carbon skeleton in this cysteine, the carbon skeleton is coming from serine and the sulfhydryl is coming from methionine. So the carbon skeleton in cysteine synthesis, it is coming from serine. And the sulfhydryl, it is coming from methionine. Okay, now what happens? This homoserine, it then changes into alpha ketohydroxybutyrate, which then changes into propenyl CoA, and this propenyl CoA then has glucogenic phase. Okay, it will be converted into succinyl CoA and it will enter gluconeogenesis, acts as a substrate for gluconeogenesis. So that was about synthesis of serine, and also we see the glucogenic rate of homocysteine. How your homocysteine will combine with serine to form cystathionine, then that cystathionine split into homoserine and cysteine. And this is transsulfuration reaction, because when you use cysteine, this carbon skeleton is coming from serine and the sulfhydryl group, it is coming from methionine. Now talking about the deficiency of PLP, that is vitamin B6. PLP, it was needed in the formation of cystathionine, as I told you. So it lead to increased homocysteine and homocysteine. Now the first reaction where your homocysteine was combining with serine, that will not take place. Now what will happen? Why will it not take place? Because the coenzyme PLP is not there. So there is deficiency of PLP. So the first reaction, cystathionine will not be formed. So therefore your homocysteine will be increased and its accumulation will lead to the formation of homocysteine. That is, two homocysteine will join together via disulfide bond to form homocysteine. So in case of deficiency of PLP, there is increased homocysteine and homocysteine. And also we've already seen that in B12 and B9 also there was homocystinuria seen. Okay. A B6 deficiency can also lead to coronary artery disease and cerebrovascular accidents due to increased homocysteine. Now, cysteine synthesis, we've already seen that how cysteine is derived from homocysteine and serine. Now, clinical correlations. These are very important, and in these also, classic homocystinuria happens to be the most important. That is what we'll be taking in detail. Rest of them, I've taken only one-liner from the point of MCQs. Okay, so the first one, Otthouse syndrome, also known as Smith-Strang disease. There is defect in methionine transporter in intestine leading to defective methionine absorption. So it's saying that, let's suppose this is your intestine. So the methionine in the intestine, it is not absorbed because the transporters, there's a block in these transporters, they are defective. This is not taking place, okay? There's no absorption in the blood of methionine because the transporters in the intestine, they are defective. Now, primary hypermethionemia, there's defect in methionine adenosyl transport. That is, MAT enzyme is defective. Characteristic feature, you have boiled cabbage odor. Now, classic homocystinuria, there's deficiency or defect in cystathionine beta synthase. So, homocysteine and serine will not join together to form cystathionine. Now, cystathionuria, this is because of defect in cystathionase. Non-classic homocystinuria, it is due to defect in methyl tetrahydrofolic acid and methyl cobalamin. And cystinuria, it is part of Geroid's tetrad, and we've already discussed what other inborn errors of metabolism are included in Geroid's tetrad. I even told you a mnemonic for it, CAP. CAAP, cap with double A. So, cystinuria, alkaptonuria, albinism, and pentosuria, they are included in the Geroid's tetrad. And your cystinuria, it is one of the most common inherited disease. Cystinuria is identified by cyanide nitroprusside test. Okay, cyanide nitroprusside test. Okay, and also it is the most common inherited disease. Okay. Now, what is the defect? The defect is in amino acid transporter in intestine and renal tubule. So, the transporter in renal tubule for reabsorption of these four amino acids is defective. That is, your cysteine, ornithine, lysine, and arginine. They are excreted in the urine. Okay. Now, let's talk about classic homocystinuria in detail. This is important. So the clinical features include there's initially it is asymptomatic, then there's decreased vision. It's usually seen as the first symptom with the child develops at the age of 3. There's progressive myopia, there's quavering of iris known as irodonisis, then ectopia lentis, that is there's dislocation of the lens. It is usually dislocated medially and downwards. Then there's developmental delay, poor scholastic performance, skeletal deformities. Then pectus excavatum, that is sternum is pushed inwards, and pectus carinatum, that is sternum pushed forward. Then you have later on they can also develop thromboembolism. Now talking about the biochemical defect. The defect is in cystathionine beta synthase. Therefore, homocysteine cannot combine with serine to form cystathionine. So that is the first enzyme which was requiring PLP that is defective. Now there is increased homocysteine in the blood, there's increased homocysteine in urine, and there's decreased cysteine synthesis. Why will be cysteine synthesis decreased? Because obviously the cystathionine is not being formed, and there's normal methionine levels because your homocysteine can still go for methionine regeneration. So methionine levels will be normal. Now treatment includes high dose of vitamin B6. Why will you give high dose of vitamin B6? Because your B6, that is PLP, is the coenzyme for defective cystathionine beta synthase. So whatever residual enzyme activity is there, it can be activated by giving PLP. Now restriction of methionine with cysteine supplementation. Also, you will restrict methionine because your methionine can be regenerated from homocysteine. Okay, so you only have to supplement cysteine. Then betaine, that is trimethyl glycine supplementation. This is a new treatment. So what is happening? It has trimethyl glycine. Okay, so your homocysteine should be betaine, that is methyl. Okay, so what will be formed? Methionine is formed. Okay, so it helps in remethylation of homocysteine, which is responsible for all the symptoms. So what is happening? Those symptoms that are basically occurring because of homocysteine, they will not be present because your homocysteine, it will be remethylated to form, to regenerate methionine. A non-classic homocystinuria, it is of two types due to defect in formation of methyl tetrahydrofolic acid and due to defect in formation of methylcobalamin. Okay, so here the enzymes are not defective, but rather what is happening, there is defective formation of methyl tetrahydrofolic acid. So methyl tetrahydrofolic acid is formed by an enzyme known as methylene tetrahydrofolate reductase. So this methylene tetrahydrofolate reductase, that is defective. See, what was happening? Your homocysteine, it was regenerating methionine. Okay. Now for this regeneration, what was happening? Methylation has to take place. Okay. And for this methylation, what was happening? You needed methyl tetrahydrofolic acid was the donor, but it cannot directly donate to homocysteine. So it was donating to what? It was donating it to cobalamin. So what was being formed? Methyl cobalamin was being formed. Methyl cobalamin was formed. And your methyl tetrahydrofolic acid, it was changing into p-tetrahydrofolic acid. Now what is happening here? What is defective? This methyl tetrahydrofolic acid. This is formed from methylene tetrahydrofolate. And the enzyme is methylene tetrahydrofolate reductase. Now here this formation is defective. Okay, this is exactly what it says, that methyl tetrahydrofolic acid is formed from an enzyme methylene tetrahydrofolate reductase, which is defective. So here this enzyme is defective. So your methylene tetrahydrofolic acid is not formed. So this is non-classic homocystinuria. Now, the other is it can be due to defect in formation of methylcobalamin. Thus, homocysteine cannot form methionine. So, what is happening? The methionine will be decreased, and but normal level of cysteine will be seen. Megaloblastic anemia is there to differentiate between two non-classic homocystinuria. Okay, because your megaloblastic anemia, it is present in methylcobalamin defect and absent in methyl tetrahydrofolic acid reductase. Okay, so specialized products that you get from cysteine, that is cysteine decarboxylation, give beta mercaptoethanol. Coenzyme A, it also has cysteine, so the sulfhydryl group of cysteine in coenzyme is highly reactive. Then taurine, that is also derived from cysteine, it conjugates bile acid. We've seen it from styrofoline and all that. Then glutathione, it is involved in free radical scavenging. Then your cysteine, cysteine is formed. How? Your two cysteine, they join together by two sulfhydryl groups forming a disulfide bond, which leads to the formation of cysteine. So that was about the sulfur-containing amino acids, that is cysteine and homocysteine. Moving on to branch-chain amino acids, very important, especially the clinical, that is maple syrup of branch-chain amino acid, that is very important, okay? So let's begin with branching amino acids. The examples are your leucine, which is purely glucogenic, isoleucine, which is both glucogenic and ketogenic, and valine, which is purely glucogenic. Now, all branched chain amino acids, they are non-polar and they are all essential because the synthesis of branched chain amino acids, they require more than 7 enzymes. So the body is not going to spend that amount of energy to synthesize all those branches. Okay, rather it will prefer to have it through diet. Now, three basic reactions for all branching amino acids. They are transamination, oxidative decarboxylation, and FAD-dependent dehydrogenation. Okay, so these are the general reactions, the three basic reactions. What is happening is, firstly, your branching amino acids, they are undergoing transamination. So on transamination, obviously, what is formed, you all know, the keto acid is formed. Okay, so the corresponding keto acid for that branching amino acid is formed. The coenzyme we know for transamination is PLP. Now what is happening? There is oxidative decarboxylation taking place. So the branching keto acids that are formed, they undergo oxidative decarboxylation. That is removal of carbon dioxide. So by releasing one carbon dioxide, it becomes a corresponding branch chain acyl-CoA, and the enzyme is branch chain keto acid dehydrogenase. Very important, okay? This enzyme, and then this is followed by, now the branch chain acyl-CoA, it undergoes FAD dependent dehydrogenation, and the enzyme is acyl-CoA dehydrogenase, and depending on the amino acid, we get different products. Okay, so here taking a look at this, at these reactions, okay, not very important, but for the sake of understanding, you should know. If you know the general reaction, that is more than enough to have a branch chain amino acid. Here in this case, we're taking the example of leucine, okay? So now this leucine, it is undergoing what? It is undergoing transamination. Okay, and for transamination, the coenzyme we know is PLP. So transamination of an amino acid, it will form what? A keto acid. You will end up with a keto acid. Here in this case for leucine, the keto acid is alpha keto isocaptoate. You see your amino group, it has been removed and you end up with a keto acid. You can see here. Now what happens? Then this undergoes oxidative decarboxylation and you can see carbon dioxide being removed. Decarboxylation taking place. Okay. So what is happening after decarboxylation? You end up with acyl-CoA. Here in this case, it is isoviryl viryl CoA. Then what happens? FAD dependent dehydrogenation takes place. So an FAD dehydrogenase enzyme acts and you end up with different products depending upon the amino acid. So in case of leucine, you end up with methyl carotenoid CoA. In case of valine, you will have methyl acryl CoA. And in case of isoleucine, you will have tiblyl CoA. These names are not important. You should just know that any branch chain amino acid, it will first undergo transamination to form the corresponding keto acid. Then it undergoes oxidative decarboxylation to form the corresponding acyl CoA. And then undergoes FAD dependent dehydrogenation to form different compounds. Okay, so that is important. And the enzyme for decarboxylation, that is branching keto acid dehydrogenase here, this is important. Okay, because with clinical is based on the defect of this enzyme. Okay, and learning these reactions in the substrate in the product, this is never really asked and not very important. Okay, yeah, but talking about the glucogenic and the ketogenic phase, this here, it will enter ketogenic pathway. And this will form propionyl CoA. And we know this propionyl CoA, it enters glucogenic pathway. And this here, dialyl CoA, it forms propenyl CoA and also acetyl CoA. So your propenyl CoA will enter glucogenic pathway and your acetyl CoA we know is ketogenic. Therefore, we say so isoleucine is both ketogenic and glucogenic. Okay. Moving on to branch chain keto acid dehydrogenase. So this is a multi-enzyme complex similar to pyruvate dehydrogenase. It is a multi-enzyme complex. So the three enzymes that are present in it are branch chain keto acid decarboxylase, known as E1 enzyme. Then you have dihydrolipoyl transacylase, that is E2, and dihydrolipomide dehydrogenase E3. And the coenzymes are coenzyme A, thymine pyrophosphate, which is associated with an E1. Lipomide, you have PAD and NAD. These are the coenzymes. Now you branch in keto acid decarboxylase, that is the E1 enzyme. It is a product of two genes, that is E1 alpha gene and E2 beta gene. And your E2 enzyme, it is a product of E2 gene, and your E3 enzyme is a product of E3 genes. So your branch chain keto acid dehydrogenase, it is a multi-enzyme complex made of three different enzymes. And then we've spoken about that what are the genes for these three different enzymes. Now, any defect in any of the genes will lead to a defect in the particular enzyme, and what you will end up with a disease known as maple syrup urine disorder. Okay, so it says, so the three gene products are forming this multi-enzyme complex, that is branching keto acid dehydrogenase, and each gene defect is associated with a disorder known as MSUD, that is maple syrup urine disorder. Now your type 1A defect, it is because of E1 alpha defective, and it is the most common MSUD. Now, type 1B, it is because of E1 beta defective. Type 2 is because of your E2 gene being defective, and type 3 because of E3 being defective. Now, talking about maple syrup urine disease. The introduction about it, it is usually seen in neonates, okay? So, the disease is going to start in the first week of life. Then there is defect in branch chain keto acid dehydrogenase. So the defect is in oxidative decarboxylation. So we've already seen after transamination what was happening. Oxidative decarboxylation was taking place in the enzyme of BCKD. So this oxidative decarboxylation will be affected now. As a result, branch chain keto acid will accumulate in corresponding branch chain amino acid accumulates. Okay, so we know branch chain keto acid, it was undergoing oxidative decarboxylation to form acyl CoA. Now here, this enzyme is defective. So your branching keto acid, this is going to accumulate, and also the corresponding branching amino acid will increase. Now, the clinical features include, it is usually present in neonates with feeding difficulties. There's failure to thrive, there's lethargy, there's acidosis, convulsions, there's hypotonia, that is decreased muscle tone with bouts of hypotonia. Urine on refrigeration for a night gives a sweet smell of maple syrup, that is burnt sugar in the morning. So this is why it has its name, because the urine, it gives a smell of burnt sugar, which is known as maple syrup. Now, diagnosis: increased branch in amino acid and branching keto acid present in urine. Now, talking about this test, it can be tested by dinitrophenylhydrazine test. A peculiar yellow color precipitate is obtained in this case, and in Rothera's test, a purple color ring is obtained at the junction. And treatment includes, you have to restrict the diet having branch in amino acid, and there's supplementation with thymine, because decarboxylation of branching keto acid, it requires thymine. Okay, so decarboxylation, it requires thymine. Now, another clinical apart from maple syrup is isovaleric aciduria. There is defect in leucine catabolism. The enzyme defective is isovaleric acid dehydrogenase. It is a fat-dependent enzyme. It has a peculiar smell of sweaty feet. Okay. So there is severe metabolic acidosis and neurological defects are seen in isovaleric aciduria. So that was about branch chain keto acid. Mostly what you asked here is maple syrup urine disorder, very important and frequently asked. So that is what you need to know from branching keto amino acids. Now moving on to basic amino acids. Okay. So basic amino acids, they are positively charged because we all know they have extra amino groups. So which is going to provide its positive charge, and they include your histidine, arginine, and lysine. Now let's take the introduction, the structures of histidine, arginine, and lysine one by one. So your histidine, it is semi-essential, it is polar, it has a imidazole ring. So you need to know, sometimes in MCQs you get asked histidine has a, and you have options like imidazole ring, guanidinium group, epsilon amino acid and all that. So you need to know that histidine, it has a imidazole ring. And what is that? It is a 5-membered ring with 2 nitrogen. So as you can see here in this diagram, this here a 5-membered ring with 2 nitrogen. So this is your imidazole ring. Okay, and it is glucogenic. We're talking about arginine. It is essential under certain conditions. It becomes semi-essential. It is more basic. Okay, it is a polar amino acid and just most basic. Why most basic? Because it has a maximum number of amino groups. Because it has maximum number of amino groups. Now it has guanidinium group, that is 3 amino groups attached to the carbon. So, and also it is glucogenic. So if you here have a look at the structure, this is the guanidinium group 1, 2, and 3, 3 amino groups attached to the carbon. So this forms your guanidinium group. Okay. Moving on to lysine, it is an essential polar amino acid and it has epsilon and genic. If you see the structure beta, delta, gamma, and then you have epsilon. So to this epsilon, you can see an amino group is attached. So we say it has epsilon amino group, and this is your lysine. Now moving on to histidine metabolism. So if you talk about histidine metabolism, so biologically important amine, so your amino acid that is histidine on decarboxylation, it is giving histamine. And this histamine, it is involved in allergic reactions. I'm sure you must have studied about this in physiology. So you see histidine on decarboxylation, that is carbon dioxide is removed, it is going to give what? Histamine. Okay, and this histamine, the major cells that are producing histamine, they are your platelets, mast cells, and basophils. Okay, so your histamine, it is majorly produced from platelets, mast cells, basophils. Okay. Now, histidine is it is also acted upon by an enzyme known as histidase to form uroconate, which through a series of reactions ultimately form alpha-ketoglutarate. And we all know that alpha-ketoglutarate, it has a glucogenic state by entering TCA. So let's have a look here at the reactions. What is happening? This is your histidine, and then this histidine, it undergoes non-oxidative deamination. So what do you get? You get uroconic acid, okay? That is non-oxidative deamination. So amino group is removed, and the enzyme here is histidase enzyme, okay? Now what happens? This uroconate that is formed, it is acted upon by another enzyme known as urokinase to form imidazolone propanoic acid, okay? And what is happening in this reaction? You can see water is being added. So your uroconate, it undergoes hydration. It is getting hydrated to form imidazolone propanoic acid. Then this is hydrolyzed by an enzyme known as imidazolone propionate hydrolase to form what? Formaminoglutamic acid, that is FIGLU, an important intermediate in histidine metabolism. Okay. Now this FIGLU, it is cleaved into glutamic acid and formamino tetrahydrofolic acid. Okay. So here the tetrahydrofolic acid, it is the acceptor of formamino group, as you can see. Folic acid here. Folic acid is the acceptor of formamino group. Okay, so on cleavage, the glutamic acid that you get, that glutamic acid, it undergoes transamination to form alpha-ketoglutarate. Alpha-ketoglutarate. And we know that alpha-ketoglutarate, it then enters glucogenic pathway to form glucose. So that was about histidine metabolism. First is that it can undergo decarboxylation to give histamine, which is involved in allergic reaction. And the second metabolism, its metabolic fate is that it is acted upon by an enzyme known as histidase to form uroconate, and then that uroconate undergoes several other reactions to finally form FIGLU, that is formaminoglutamic acid. Then that FIGLU is cleaved to form glutamic acid and formamino tetrahydrofolic acid. So the acceptor for formamino group is your folic acid. Okay, your glutamic acid then will undergo transamination to form alpha ketoglutarate, which then forms glucose. So what are the key points to remember? That FIGLU is an intermediate of histidine. Like if you remember, kynurenine, I told you, was an intermediate in tryptophan metabolism. FIGLU is excreted in urine and folic acid deficiency. Why? Because if there is folic acid deficiency, your formamino group there will not be an acceptor for formamino group. Okay, so in that case, what will happen? Your FIGLU will be accumulated, and that FIGLU is excreted in urine in case of folic acid deficiency. Then you have histidine load test. What is that? It is done to assess the folic acid status in the body. Okay, so what is happening in the histidine load test? You are assessing the levels of folic acid in your body. So what is happening? Lots of histidine is given, and in case there is tetrahydrofolic acid deficiency, your FIGLU will be excreted in the urine. Okay, why? Because there is an overload of histidine and there is an actual deficiency of folic acid. What will happen? Again, there will be no acceptor for formamino group on cleavage of FIGLU. So what will happen? Your FIGLU will not be cleaved. FIGLU will start accumulating and it will be excreted in the urine. So that is histidine load test. Moving on to the products that we get from histidine. So it is carnosine, anserine, and homocarnosine. What is your carnosine? It is beta-alanine plus histidine. So when beta-alanine combines with histidine, you end up with carnosine. What is anserine? It is beta-alanine plus methyl histidine. So it is methylated carnosine. And these that is carnosine and anserine. It is present in the muscles. It is present in muscles. Okay, and your homoserine, which is GABA plus histidine. It is present in brain. And all of them. So you see, whether it be your carnosine, anserine, homocarnosine, all of them, they contain histidine, as you can see. Now talking about arginine, the metabolic functions of arginine include use in the synthesis of agmatine, that is, which is hypertensive, then used in the synthesis of keratine. We've already seen this. Then used for the synthesis of urea. You already spoken about this in urea cycle. In keratine, we had seen how your keratine is synthesized from glycine and arginine and methionine, if you remember when we were talking about methionine and glycine. Then it is used in the synthesis of ornithine, and it is used in the synthesis of nitric oxide. Now, what is talking briefly about nitric oxide before I end the lecture? It is important, your nitric oxide, you even get asked about this in Vivas, then otherwise also the role of nitric oxide, especially the functions are very important, okay? So what is it? It is an endothelium derived relaxing factor. It is a free radical, that is, it has unpaired electrons, so it is highly reactive. Highly reactive. Because it has unpaired electrons. Then it is a gaseous molecule, very short half-life. Nitric oxide is formed from arginine by the enzyme nitric oxide synthase. This point is important. So you see, this is your arginine. So arginine is acted upon by an enzyme which is known as nitric oxide synthase, NOS, and you end up with nitric oxide plus citrulline. So this is how your nitric oxide is formed. Now talking about this nitric oxide synthase enzyme, it is a monooxygenase and it contains hemes. The enzyme is not very important. You rarely get asked about it. But briefly speak about it. It has 5 cofactors, that is heme, tetrahydrobiopterin, NADPH, FMN, and FAD. It has 3 different isoforms. Now the functions of nitric oxide, this is important, okay? So you need to know the functions of nitric oxide. So speaking about that, it is a vasodilator. It is important in penile erection. So being a vasodilator, it relaxes smooth muscles in the corpus cavernosum and increases blood flow into the penis and makes it erect. So that was one function. Now it also acts as a neurotransmitter. It is used in the treatment of pulmonary hypertension. It produces pulmonary vasodilation without lowering the systemic blood pressure. It is used in the treatment of impotence as sildenafil. So what is happening to sildenafil? It is going to inhibit cyclic GMP phosphodiesterase. So your cyclic GMP phosphodiesterases, what are they doing? They are involved in the degradation of cyclic GMP. So they degrade cyclic GMP. Okay, and your cyclic GMP, if the inhibitors, if the phosphodiesterases which are degrading the cyclic GMP, they are inhibited. Okay, so if the cyclic GMP phosphodiesterases, they are inhibited, your cyclic GMP will no longer be degraded. So what will happen? Your cyclic GMP levels will increase. And your cyclic GMP is the second messenger of nitric oxide. That is, your nitric oxide acts via cyclic GMP. So nitric oxide acts via cyclic GMP. Also, this can form an important MCQ. That is, what is the second messenger of nitric oxide? That is cyclic GMP. And then glyceryl trinitrite, it is used in the treatment of angina pectoris. Because of glyceryl trinitrite produces nitric oxide which dilate coronary artery. Okay, so that is why it is used in the treatment of angina. Now talking about lysine, not very important, never really asked. Talking about the metabolic functions, histones are rich in arginine and lysine. So only from the end of MCQ, I'm going to discuss few facts. That is your histones. They are positively charged and they are rich in arginine and lysine because they have basic amino acids. Okay. Now, lysine on putrefaction by bacterial enzymes, it produces cadaverine, which is a polyamine. And then your carnitine, it is also derived from lysine and methionine. If you remember your carnitine, it was a fatty acid transporter. The fatty acid, it cannot be transported to mitochondria for beta oxidation if there is carnitine deficiency. Okay, so that is the importance of lysine. That is, your carnitine is derived from lysine. Then your histones, they also have lysine. And then lysine on putrefaction by bacterial enzymes, it produces cadaverine. Okay. How? Your lysine, it will be undergoing decarboxylation to form cadaverine. Okay. So with this, we come to the end of the class and also to the end of amino acids, each of them and their metabolism. Thank you for watching. If you have any queries, you can write them in the comments. So we're done with the major chunk, actually not the major chunk, but with the entire metabolism. We're finished with carbohydrates, lipids, and amino acids. Okay. Thank you for watching. See you in the next class.